Two more micro lectures for 12. The last one's the easiest because it's just the synapse. This one's going to look at the electrophysiology and how does an action potential form, and then how do we get that signal down the neuron. So what's the electrical change difference? How stimulation of a neuron causes local electrical response in its membrane, how the local response generates a nerve signal, and how a nerve signal is conducted down the axon. Very complicated. So Galen, we talked about him. He thought the brain pumped a vapor called physic-pneuma through hollow nerves and squirted it back into the muscles. You might have remembered Aristotle thought the brain was just like a radiator for blood to give off heat. René Descartes, 17, supported this theory. So even in the 17th century, they didn't really know what the heck the brain did. And then Galvani discovered the role of electricity in muscle contraction in the 18th century. And he did that by poking things, pretty much. So he started poking things and seeing how things moved. Camillo Golgi, him and Ramon Cajal after him, they did something very interesting. They used silver nitrate. It says staining neurons with silver. But there's this chemical called silver nitrate. All you do is you take a slice of tissue, you put silver nitrate on it, and what it does is it just kind of makes all of the cell tissues visible. So you can see its outlines, you can see everything like that. And the thing that he saw, and Ramon Cajal after him, he saw that there's a neuron, and there's a space between the neuron and the cell after it. And this got people thinking that, yeah, it might be chemical and electrical. So it wasn't until the end of the 19th century, beginning of the 20th century, where they finally started saying, you know what? It's not just a wire all the way going to the cell. It's a wire or tube, and then there's a gap called the synapse. So now what we look at the neuron generating the electrical signal, and then how does that electrical signal at the synapse release a neurotransmitter. So electrophysiology is the cellular mechanism for producing electrical potentials and currents. The electrical potential is the difference in concentration, charged particles between one point and another. An electrical current is the flow of charged particles from one point to another. What you need to know: sodium, potassium. Sodium and potassium are either going to flow into a cell or out of the cell. Your job with this lecture is to figure out, is the sodium that goes in or is the potassium that goes in? And think about it. Electrical potential is just a difference between charges. So if you're pumping positive charges out of the cell, what does that make the cell? It'll make it more negative. It goes back to chemistry of life when we talked about electrons. If you're moving positive out, the cell becomes more negative. If you allow positive things to flow back in, it makes the cell more positive. That's the basic theory we're going to be talking about for the rest of this lecture. So all living cells are polarized. They have a different charge between the inside and outside of the membrane. That difference is called the resting membrane potential. That's the difference between the outside and the inside. And in most cells, it's around 70. Some neurons can be 90, some cells can be in the hundreds, but for the most part, we'll just arbitrarily say negative 70 is our resting unstimulated neuron, just sitting there resting. And it being negative means there's more negative particles on the inside than on the outside. So because there's more negative on the inside and on the outside there's more positive, that's why we have that unequal electrolyte distribution. So the resting membrane potential results from the combined effect of three factors. Ions can just diffuse down their concentration gradients into the cells, because remember from when we talked about the cell membrane, you might have a channel where ions can just flow through it. You also are going to have plasma membranes that allow some ions to go through, other ions cannot. Then finally, some of these are going to be attracted to each other. Positive is going to be attracted to negative. So how does the body make a resting membrane potential? Potassium ions, greatest influence on resting membrane potential. Write that down and memorize it. Potassium, the plasma membrane is more permeable to potassium than any other ion. So potassium can flow in or out. What we're going to do, we're going to figure out right now if it's flowing in or if it's flowing out. It's going to leak out of the cell until the electrical charge of cytoplasmic anions attract it back in. An equilibrium is reached, and then potassium stops. Potassium is about 40 times as concentrated in the intracellular fluid as it is in the extracellular fluid. So if it's more concentrated inside, it's going to diffuse down its concentration gradient out of the cell. All right. So cytoplasmic anions cannot escape because they're big and they're charged. Phosphates, sulfates, small organic acids, ATP, proteins, RNA, they kind of are in there and they don't really like to leave. And anions are negative. The membrane is not so permeable to sodium. So sodium is found outside the cell. What happens is that sometimes it leaks and diffuses down its concentration gradient into the cell. And sodium is about 12 times as concentrated on the outside of the cell as it is inside of the cell. And the resting membrane potential is less permeable to sodium than it is to potassium. Just to, say it again, potassium is the main one. You have a very special pump inside each one of your cells. It's called the sodium-potassium pump or sodium-potassium exchange pump. What it does, it pumps out 3 sodium and it brings in 2 potassiums. And if you kind of sit there, you're like, it's pushing out 3 positive, only bringing in 2 positives, that's going to make it more negative. The majority of your energy, your ATP, is made just to create energy for that pump. Depending on kind of, what book you kind of look at, around 70% of all your energy in your body every single day is going to be required for the nervous system. And of that 70% for the nervous system, the majority of that energy is just going to this sodium-potassium exchange pump. And then it needs glucose and oxygen to be supplied to the nerve system or nervous tissue, because oxygen and glucose is needed for ATP. And this is very, very, very important to making the resting membrane potential. So this is sodium, that's potassium. These are just channels. You can see in this diagram, sodium is the orange thing on the outside. There's a lot of sodium outside, so it kind of leaks into the cell. Potassium, very concentrated inside the cell. It leaks out of the cell. Now you know why sodium is concentrated outside the cell and potassium is concentrated inside the cell, because of that sodium-potassium exchange pump. So what's a local potential? This is a small localized, so in the same vicinity, disturbance in the membrane. So when a neuron gets stimulated, something in a small area gets disturbed or receives a stimulus. Then, this is always going to begin at the dendrite, because this is where the signal is going to come in. It's going to go through the soma, travel down the axon, and then end in the synaptic knob. But there's several steps it needs to do to get that local potential all the way down the synaptic knob. So neurons can be stimulated by chemicals, light, heat, mechanical disturbances. If it's light, it's got to be a photoreceptor in the retina. If it's going to be mechanical disturbances, we'll talk about a stretch receptor where your muscle stretches and it sends a signal saying, hey, I'm stretching, stop doing that. Or heat or light and other types of receptors will create different types of potentials. This happens, it opens sodium gates and allows sodium to rush into the cell. If sodium rushes into the cell, does the cell become more positive or negative? Yep. Positive goes into the cell. It's now becoming more positive. Perfect. Voltage across the membrane drifts towards 0 or gets closer to more positive, because remember, it starts at -70, so it's becoming more positive, and -60 is more positive than -70. That's called depolarization. So when membrane voltage shifts up or more towards positive or less negative, depending on how you want to word it, that's called depolarization. Sodium diffuses for a short distance on the inside of the plasma membrane, producing a current that travels towards the cell's trigger zone. The short range changes in voltage is called the local potential. Because if you think about it, sodium is just going to go into one little part of the membrane. What's happening right next to it? Nothing. Until that travels over here, then that can start to open sodium channels, and then that local area becomes depolarized. So that's why when you're thinking about local potentials, they're very, very, very small areas on dendrites or the soma. Now, you have something with these potentials, they can be called graded, which means they can vary in magnitude. If you have one channel open, that might not change that much. You have hundreds of sodium channels open, that's going to be a greater change. So you can have a small magnitude or a large magnitude, and that magnitude just opens more sodium gates. It can also become decremental, which means the further away they get, the smaller they become. Same thing here, like if I hit a table here, the further you get away, the less sound or less vibrations you feel. They can be reversible. So pretty much when they stop, sodium or potassium will start to diffuse out and bring it back to its resting membrane potential. You can have excitatory or inhibitory. What if I said, so it says some neurotransmitters, glycine, make a membrane potential more negative and hyperpolarize it. How would you make a cell more negative? What type of channel would you open up? Something with a negative charge. And chlorine has a negative charge. So if you open a chlorine channel, chlorine goes into the cell, more negative on the inside of the cell, it starts to become more negative. That's called hyperpolarize. Becomes more negative. So if you have something that's inhibitory, it's more than likely opening a chloride channel that makes a cell more negative. If it's excitatory, it opens a sodium channel, makes it more positive, for the most part. And here you can kind of see sodium opens, sodium goes in, and it just becomes one local area on that dendrite. How do we get to an action potential? So the more dramatic change produced by voltage-regulated ion gates in the plasma membrane, the more you have, the more potential you have for that to reach the trigger zone or the axon hillock, and then that can create your action potential. It only occurs where there is a high enough density of voltage-regulated gates, which means that in the axon hillock, you have gates that are triggered by voltage. So these specialized gates are going to be here in the axon hillock. So when all of that potential, that ion change gets to that area, it's going to open another type of channel. And we'll talk about that in a little bit. So the soma doesn't have a lot of these gates, so it can't produce an action potential. The trigger zone, you can see how many more of those gates they have. So that's where the action potential is going to happen. If excitatory local potential spreads all the way to the trigger zone or the axon hillock, and it's still strong enough when it arrives, it's right at threshold, it's going to open these gates and generate an action potential. Action potential is a rapid up and down shift in membrane potential. Sodium ions arrive at the axon hillock, depolarize the membrane at that point. That opens voltage-gated ion channels, and if it's at threshold, which is -55 for this hypothetical cell, it's going to create an action potential. When threshold is reached, axon fires, produces an axon channel. The more sodium channels open, is sodium going in or out of the cell again? Sodium's going into the cell at this point. In the trigger zone, in a positive feedback cycle, creating a rapid rise in membrane voltage. That's called the spike. What do we have when the resting membrane passes 0 millivolts? Sodium gates become inactive. They now close. They begin closing, but because they don't close right away, voltage is going to peak right around +35. Membrane is now positive on the inside and negative on the outside. This now means that other gates are going to open and cause depolarization. So when the potassium gates open, they're going to flow back into the cell, they're going to flow outside of the cell to repolarize the cell. So potassium is repelled by the positive intracellular fluid and exit the cell. Their outflow repolarizes the membrane and the cell and shifts the voltage back to negative numbers. Potassium gates stay open longer than the sodium gates. That's going to cause something called hyperpolarization. And pretty much what we're going to do, look at the action potential. So this is everything we talked about. Resting membrane potential. This is at sodium-potassium exchange pump. Sodium gates open slowly. There's your graded response, getting more positive. Right when it reaches threshold, voltage-gated sodium channels will open. Will cause depolarization. They close. Voltage-gated potassium channels open. They flow out of the cell, repolarize the cell, and because they stay open a little bit longer, it goes down past resting membrane potential and then comes back up. So if you think that's complicated, it is. Why physically or physiologically, why the potassium channels stay open longer and why they're open all the way down here? This makes sure that an action potential can only move in one direction. Because if this cell or these channels right here went through an action potential, the one next to it would have to go to the one that's already at resting membrane potential, not the one that's a little bit lower. It's all or none. Right when you reach threshold, the voltage-gated sodium and voltage-gated potassium channels open and close. So you can't go back. You can't stop it once it already starts. It doesn't get weaker with distance. It's irreversible. Once started, it cannot be stopped. This is what happens. Sodium gates will open to, sodium and potassium gates are closed at resting membrane potential. That's why you have the sodium-potassium exchange pump. A couple of sodium channels will open right when it reaches past threshold. voltage-gated sodium channels open, allows sodium to rush in, more positive on the inside. They start to close right here, where we get the potassium gates open. They start to flow out of the cell and repolarize the cell. And because they go slower, they go a little bit past resting membrane potential. So you have absolute and relative refractory period. Can a neuron fire if it's in the absolute refractory period? And you only have two gates to use. You can either open your voltage-gated sodium gates or voltage-gated potassium gates. During the absolute refractory period, those two gates are already being used. So even if you really wanted to send another signal, you would have to wait until after this signal is done. The relative refractory period, look at how much further away from threshold it is. In the relative refractory period, could you still send a signal? Yeah. You would just need more input to get it back up to threshold. So in the relative refractory period, because we're more negative, we're going to need more positive ions to flow back into the cell to get us closer to threshold. So if you're still in that relative refractory period, you can still fire, but it's going to take more input. In absolute, can't fire. Relative, it can, but you just need to have more positive ions flow into the cell so it fires again. So, conductive of nerve signals, this is for communication to occur. The nerve signal must travel from one end to another. Unmyelinated fiber has voltage-regulated ion channels along its entire length. Unmyelinated means that if this one opens and closes, the one right next to it has to open and close, open and close, open and close, open and close. You get the point. So it'll take a lot longer for all those ions to flow in and out of the axon. But in myelinated, they can skip. So we'll talk about that in a little bit. And the action potential from the trigger zone causes sodium to enter the axon, to fuse into adjacent regions beneath the membrane, and that's going to be the goal of myelin and insulation. So when we look at this, we're still going to have sodium, we're still going to have potassium open and close, but we're also going to have that hyperpolarization from the potassium that we need to memorize. And then this opening and closing is going to be the way it travels down the axon. It's going to happen via a chain reaction. So here you go. So action potential is right here. It's positive on the inside, negative on the outside, and then you're going to be able to go to the next one next to it. That'll then be the action potential, and then travel down to the next side that's the action potential. And you can see, because of that relative refractory period, because it's hyperpolarized, that's the reason why, and the physics behind why a nerve impulse just travels in one direction down the axon. Aren't you glad that wasn't in red? You should be. So, voltage-gated channels needed for action potentials, fewer than 25 per micrometer, and myelin-covered regions up to 1,200,000 per micrometer in nodes of Ranvier. All that means, and if it's myelinated, you don't have many in between. You have them all between the nodes of Ranvier. So instead of it just kind of going right next to it and traveling down that way, this is called saltatory conduction. And what happens is it looks like it jumps from node to node. So instead of having them right next to each other, they start to jump and jump. And that's how a myelinated axon can conduct a signal much quicker than a non-myelinated one.